So the normal cotton variety was affected by this insect, and this insect is known as cotton wall worm, right? So what approach we have used for it? There is one bacteria, and this bacteria is known as Bacillus thuringiensis. Bacillus thuringiensis, right? In short, BT. So this bacteria has one specific gene. We call it BT gene. What we call it? BT gene. And there are variety of BT gene. We will come to it. So first let us call it BT gene. So, this BT gene is produce some protein. We will call it BT protein, right? And this protein when it comes in contact of the gut of insect, okay, insect is killed. So this is the main concept behind it, okay? And we have to use this idea that somehow if we are able to transfer this gene into the cotton plant, right? So then this protein will be produced by the plant, and because of that reason, this protein will enter into the insect and insect will be killed. If you are not getting it, let me repeat once again. The idea is this Bacillus thuringiensis bacteria is having a certain gene, and this gene produces BT protein, and this BT protein is an insecticidal protein. So the idea is to take out this gene from this bacteria and put it into the cotton plant. So this is what we are doing in biotechnology, right? Transferring gene from one organism to the other organism, right? So this cotton plant, we will use some vector and we will transfer it into the cotton. So cotton plant with BT gene, right? So this new improved plant is known as BT cotton. This new plant is known as BT cotton. Are you getting it? So in this plant, the parts of the plant, let's say leaf, will produce this protein, BT protein. So BT cotton plant will produce BT protein, and when the parts of the plant will be eaten by this insect, right? So when insect is feeding on this BT protein, this insect will die, okay? And what is the mechanism? BT protein, when it is in the plant, it is in form of inactive protein. It is an inactive protein, and inside the gut of the insect, there is alkaline medium. And because of this alkaline medium, this BT protein will become active, right? And insect will be killed. This will cause lysis of the gut. Lysis means breakdown of the gut, or insect will be killed. So do you have any doubt in this? Have you understood? Do you have any doubt in this? Any doubt? Any doubt, children? No? No, okay. So I will repeat it once again, okay? So what is the whole idea? Idea is we need to transfer BT gene from this bacteria to cotton plant Cotton plant will produce Bt protein. Bt protein is inactive form in the plant, and then in inside the gut of the this insect, it will become active, and then it will cause lysis of the gut of the bacteria, sorry, not bacteria, gut of the insect, and insect will be killed. Okay, in bacteria there is no gut. I don't know how I like missed this word. Okay, no problem. Now we will talk about what kind of genes are there. So these genes are known as cry gene. So we have cry1AC, we have cry2AB. So cry1AC and cry2AB, they control cotton bollworm, and cry1AB controls corn borer. Okay, here in this picture you can see this is one grown cotton ball. So this is basically a healthy cotton ball, and this is the one which has been eaten by insect. So literally there is no cotton in this ball, right? So basically these cotton bollworm are going to reduce the survival of this plant, because not survival exactly, productivity of the plant, by eating up these cotton balls. Now the another technique that we are going to use is RNA interference. Okay, or in short we call it RNAi. So if I talk about what is RNA interference or what is RNAi, basically this is a process in which we need to convert a single stranded RNA into double stranded RNA so that it will not be expressed. I repeat once again, in this process what we are doing, we are converting a single stranded RNA into double stranded in order to stop its expression in the form of protein. Okay, so what we are doing and how it will be used. For example, there are some pathogen nematode pathogen, and this pathogen affect tobacco plant. This pathogen affect tobacco plant, right? So this nematode pathogen is having certain gene, and these genes are producing a certain RNA, and this RNA is producing a certain protein, right? And this protein is the region of pathogenic nature. Right, I'm repeating once again. Okay, when one pathogen comes in our body, okay, either it will eat our organs or our cells or the body fluid, right? Different type of pathogens are there, or it will produce some protein, and this protein is going to interfere with our internal chemical reactions of the body. Okay, and this is how a pathogen causes a disease. So this is one usual mechanism, right? So from this discussion, if we eliminate this, if there will be no formation of pathogenic protein, then there will be no pathogenic nature or disease. I repeat once again, if somehow we could stop this RNA from forming the protein, then the pathogenic nature of this pathogen will not be there, right? So this nematode pathogen cause disease in the tobacco plant, right? So what we are going to do is, we will take some genes which are similar to the one which is responsible for production of this protein. So that gene we are adding into the tobacco plant. So this nematode specific gene, if I will write here, I will write it the way it is written in our book, nematode specific gene, right? So in last chapter we have learned that when a when a foreign gene is added to an organism, it start functioning if it is added to the ORI site of this particular organism, right? So this gene is added to tobacco plant. That means this gene is going to form RNA, right? And this RNA is going to form a protein. But there is one catch. And what is that? That when this pathogen will also attack this plant, it will also produce an RNA, right? And this RNA and this RNA are going to be complementary. I will just draw one structure, symbolic structure, and here also I am going to draw. Okay, so these two RNA strands are complementary of each other, right? So they will come together and they will produce one double-stranded RNA. They will produce one double-stranded RNA. Right? So this double-stranded RNA will not form any protein, no protein. And from our previous discussion, you know that this protein was the cause of disease. So when there is no protein, there will be no disease. Are you getting it? Is it clear to you? Are you getting it? Yes or no? Yes or no? Okay. So this mechanism is known as RNA interference. Okay. And from where this inspiration came? Actually, this process keep on happening in all eukaryotes, and it is one of the method of cellular defense. So this RNAi is a type of cellular defense, and it happens in all eukaryotic cell. In all eukaryotic cells, right? Understood? So what it is? RNA interference. So we can use this method for the controlling the insect pest, right? And one example that I have given is the tobacco plant, right? So the name of nematode is Meladogyn incognitia, right? I have not written the name of the nematode. It is written in your book. It is Meladogyn incognitia, right? So this was all about RNA interference. Okay, in this picture you can see there are two plants, right? This plant is having clear roots, so basically there is no infection, but this plant is the one which is infested by this nematode. So you can see there are certain swelling or cancerous growth, right? Tumorous growth everywhere in the root. Okay, so this is the condition after we use this method. Which method? RNA interference. So this is the plant after using RNA interference, and it is the infected plant without using this method, right? Now we will talk about application of biotechnology in medicine, right? Application of biotechnology in medicine. So let's come to it. Okay, so in the previous section I talked about insulin, right? So insulin is synthesized in our pancreas. right? So earlier, for the diabetic patient, when we needed insulin, we are using it, the insulin which is obtained from pancreas of pigs, right? Because pigs are being used for like pork, right? So their pancreas was used to obtain insulin. So if we compare a pig and human, right? Pig and humans. So although both are mammals, right? Both are mammals. So they are somewhat similar because pig is having insulin, humans are also having insulin. But still it is a heterologous host. Heterologous host in the sense that it is not having the same genetic composition as us. So the insulin which is coming from pig and being utilized in human is also going to have some different like different structure and composition. So this insulin which was coming from pig, right, it was producing some side effects. It was producing some side effects. So that means that to certain extent it was helpful, but it was not at all a remedy, a full-fledged remedy. The reason was that it was giving some side effects. So what we did, basically there is one company in US, okay, it is Eli, ILY, and Lilly, LILY, okay, Eli Lilly. So what they observed that there are total 3 peptide chain in the insulin. How many? 3. So there was A chain, B chain, and C chain. Right? How many chains? 3 chains. So these 3 chains are present in immature insulin, or we can call it proinsulin. So proinsulin is having these 3 peptide chain, and these are C, A, and B, right? What are these? C, A, and B. So let's say this is C, this is A, and this is B, right? Later on, when this insulin is converted, this proinsulin is converted into mature insulin. So in case of mature insulin, there are only 2 chains. And what are these? These are A and B, not the C chain. So there are A and B chain, and there are disulfide bases between these 2 chains, right? So this is the story of natural insulin. So what this company did, they synthesized A chain and B chain separately, right? And then they joined these 2 chains to produce this type of insulin, right? And now this insulin is used commercially or in clinics to treat the diabetes. So I'm repeating once again. So this is one of the application of biotech in medicine. What we did, insulin is used for treating diabetes, and earlier this insulin were being obtained from where? Pancreas of slaughtered pigs, but it was having certain side effect. If we talk about the structure of insulin, immature or proinsulin have 3 peptide chains, and mature had just 2, A and B peptide. So this company from USA, Eli Lilly, what they did, they synthesized A and B strands separately, then they introduced disulfide bases to form mature insulin, and this insulin is being produced commercially to fulfill the need for treating diabetes. Okay, so this is one of the use, right? Next we will talk about gene therapy. Next topic is gene therapy. So what is gene therapy? Let's talk about it. So in case of gene therapy, it is very simple to understand. For example, we are having nearly 30,000 genes, right? We are having nearly 30,000 genes. And we have techniques that in embryonic stage, let's say this is a zygote, right? So we can scan the DNA of the zygote and then we can find out the abnormal pieces of DNA, or those DNA segments which are going to cause some disease in the offspring, right? So you know from your this chapter reproduction that zygote is a single cell from where life start, and as this cell will grow further, it will start dividing, right? And we are having nearly 100 trillion cells, right? So the number of cell will keep increasing with the growing age, right? So we cannot change the DNA in each and every cell when this embryo is at advanced stage, right? Or it becomes a full blown individual. So what we have to do is we have to edit the zygote itself, right? So those abnormal pieces of DNA should be removed, or they should be replaced with normal functional gene or normal DNA, right? So this technique is called gene therapy, right? So this kind of gene therapy where we are editing the embryo, it is going to cure this particular person who will, I'm calling it person, it is a zygote, it will become a person. So it is going to cure this person completely, right? But there are certain procedures which are used in general. For example, if we talk about first clinical gene therapy, first clinical gene therapy. So this first clinical gene therapy was given to a 4-year-old child, 4-year-old girl child basically, okay? And this particular child was suffering from a disease called ADA deficiency. It was she was suffering from ADA deficiency, right? So the first ADA is basically one enzyme, okay, adenosine deaminase we call it. So the first step could be we can go for enzyme replacement therapy. Enzyme replacement therapy means that enzyme which is not being produced inside the body of this particular child, that enzyme we can give from outside, right? So this is enzyme replacement therapy. But the problem with this therapy is that this child has to given this enzyme periodically, like in a month or whatever the schedule is. So this child has to be given this enzyme again and again, right? But the better technique could be bone marrow transplant, right? Or another technique could be we can use lymphocytes, right? So we will take out the lymphocyte of the patient, and then we will edit the gene in the lymphocyte, and then we will infuse those lymphocyte into the patient, right? but we know that like RBC, WBC, these lymphocyte, they are having a definite lifespan. That means these cells are not going to survive, they are not going to be there forever. So this is also not a completely curative uh, therapy, right? So although there are these three methods, but these three methods are not completely curable. So the better approach is to get scan the zygote, okay, for defective DNA segments, and then it could be corrected right there in embryonic stage, and this therapy will be completely curative, right? So even if we talk about future of biotech, so they are saying that even those small illnesses, for example fever and cold, etc., these also might have some genes associated with these diseases, and it is possible that in future we might be able to edit those genes which make us sick. So the future of medicine is going to be biotechnology or genetics, where we will give therapy at genetic level, right? So we are still far from it. Let's see how far it goes, in how many years we are able to do that, but it is coming for sure, right? Okay, so this part is done.